It's like a check, check, check.
I'm going to start. There we go. We're on YouTube. Perfect. And then I'm going to start sharing my screen. There we go. All right. I can no longer see the list of attendees, but
I can see we have 42 people already.
perfect. I'm going to wait another minute or two before getting started here. Thanks for joining, everyone. So just if you're joining us, we'll get started in just a few minutes. Thanks for your patience. So if I can ask Rebecca and/or Alison to maybe just keep on top of some of the questions that are more immediate, that would be fantastic.
So Gio, I'll answer your questions. And so yeah, if you have questions, Allison and I will answer.
So while we wait here for more people to join, I'm just going to highlight a couple of features that hopefully you can see. So you're able to ask your questions either in the chat or if they are pertinent to the actual content of the talk more so. There is a Q&A button, hopefully, that you can see on the top together with the other functions there. And by clicking it, you'll be able to ask your question, and those will be recorded for us to go over at the end. And I believe you can up or down vote other people's questions as well. So I'm going to get started here. Welcome, everyone. Thanks so much for joining us. My name is Gio Rapacciuolo. I'm a research scientist in the citizen science team at the California Academy of Sciences. This is a webinar on monitoring California's coastal biodiversity with citizen science. It's a webinar that caps an 18-month collaboration between the California Academy of Sciences, the California Ocean Protection Council, and the California Department of Fish and Wildlife. And this collaboration explored and is continuing to explore the use of crowdsourced citizen science data to monitor and understand biodiversity on the California coast and assist management and policy. So just a little bit of background about myself very quickly. I joined the project 18 months ago. This has been sort of my full-time job for that time. And my background is really in big biodiversity data analysis and as an ecological modeler. And specifically, my main interest is in adapting and developing tools from macroecology and quantitative ecology and putting those at the service of conservation and management practice. So I really want to start by acknowledging the whole team here. This has been a fantastic collaboration and continues to be a fantastic collaboration. And so I highlight a few names. Rebecca Johnson and Alison Young, first and foremost, are the co-directors of citizen science here at the Academy. And they've been creating and spearheading some of the amazing citizen science initiatives on the coast here in California and beyond, much beyond. So we'll find out a little bit more about those. Luckily, they're joining us today, and so they'll be presenting a little bit about them as well. And then a lot of credit goes to OPC and CDFW for really having the foresight to see potential in these types of data and funding this incredible 18-month project here. And so I want to highlight especially Mike Esgro and Tova Handelman, who were crucial in getting this project started, and Mike Esgro, as well as Sarah Warden, who were with us the whole time, providing crucial feedback and contributing a lot to the outputs that we'll be finding out today. So let's move on. The agenda today for this webinar is divided in two parts. The first part will be sort of a broader background about the project, and we'll hear from some of our team members and provide some background about the citizen science initiatives and the data collection, as well as then aspects of analyzing those noisy data, and then some findings a little bit more in detail there. Then we'll have a short 10-minute break, and the second part will be really about the dissemination of outputs and how we went about putting outputs together, and then going over the web tools that we've put together as part of this project with a live demo there to show you how you can access the outputs of this project. And then a little bit about where next for this collaboration, and then we'll have a Q&A at the end with questions for myself and the whole team. So please line up your questions and we'll keep them all for the end. So let me get started with some broad background here. We've entered the Anthropocene era, a new geological area that's defined by the pervasive influence of humans on all Earth processes. And the big 5 drivers of biodiversity change in the Anthropocene have been described in detail here in the IPBES report that came out last year with a lot of crucial insights for biodiversity conservation. And those are, we know them, they're land use change and sea change, marine change, direct exploitation, climate change, pollution, and invasive alien species. And what I want to highlight here is that these 5 big drivers are all characterized by being global. That is, they don't stop at borders. They have a global, broad-scale geographical influence. They are dynamic. That is, they change abruptly and evolve over time. And they're ubiquitous across the tree of life. That is, with cascading effects across taxa. And here I like to give us a little bit of perspective. I like to show this cartoon that really gets at the idea of large scale. And so in 2100, the temperature change is predicted to be globally about as large. So the temperature in 2100 is predicted to be as dissimilar from today as it was 20,000 years ago, though obviously in the opposite direction. 20,000 years ago, we had a glaciation. In 2100 is predicted to be much warmer. But this really gives an idea of how large the scale of the problem is here, and how much of a challenge it will be to try and predict what might happen to living systems in those conditions. And so to to be able to understand and predict what might happen to living systems, we need information that is big in all three dimensions of ecological space. And so those are space, time, and taxonomy. And if you're big in all three dimensions or some of these dimensions, you're dealing with the domain of macroecology, large-scale ecology. And the reason that we need to be big in at least some of these dimensions is that that will allow us to explore the full range of environmental variability that biological systems have ever experienced across the whole world and throughout history. And this will enable us to derive general conclusions about how environmental change influences biological systems. So where are the big biodiversity data that will enable us to explore variation over space, time, and taxonomy? To find that out, we need to go to GBIF, the Global Biodiversity Information Facility. And GBIF is the global data repository for biodiversity. It's funded by the world's governments, and it's really the reference for biodiversity data. And it has over 1.4 billion biodiversity data points. And this brings us to the first poll, which hopefully I can bring up here. And the question I want to ask you here is, what percentage of all animal data do you think on GBIF comes from citizen science projects? Let's see. So hopefully you can see a poll. We're getting some answers. Okay, I'm gonna wait a few more seconds. It's pretty even, this. I can't even tell. Okay, I'm gonna end the polling right there. So the winner is 15%. And I'm gonna close the polling here. And 15% is pretty far. So the actual answer is here on the second line, and that's 85% of all data on GBIF come from citizen science projects. That is an astonishing number. It has to be said that this is heavily biased towards birds, and a lot of those data come from eBird and iNaturalist combined. And so you'll see that for mammals, for example, it's much closer to your answer. And for other groups, it is. So a little bit of a trick question there, but still an astonishing number. And perhaps just as importantly, 6 out of 10 of the main sort of data sets in GBIF are actually citizen science data sets. eBird is up there, and I've highlighted iNaturalist here, which iNaturalist is the tool that we use here at the citizen science team. There are our collaborators here at the Academy of Sciences. And so I wanted just to introduce iNaturalist a little bit for those of you who may not be familiar with it. And so iNaturalist is this incredible resource. It's on one hand, it's a web platform. It's a phone app that you can download on your phone. I know a lot of you have it already, but I encourage you to download it. But it's also so much more than that. It's a social network that allows people to interact around biodiversity data and allows people to vet each other's observations of biodiversity. And so the point I want to make here is that iNaturalist allows you to make biodiversity observations with just a few key bits of metadata. The first one is who you are. You have to sign up with an account. And the second one is really the main data is a picture of a living organism. And that picture gets then tagged with some geocoordinates. Your phone will give it GPS coordinates and a timestamp as well. And those coming from your phone generally are extremely precise if you take a picture from within the app. And all these three things combined, the picture and an initial ID, the timestamp, and the GPS coordinates are the basic date and type that is needed to explore biodiversity in space and time and across taxa. So hopefully after you leave today, I will convince you of this. But for now, let's just leave it at the fact that citizen science data and crowdsourced citizen science data in particular have enormous potential to help monitor and understand biodiversity on the California coast and beyond. And there are huge volumes of them. So we ought to figure out how to work with them. This brings us to this wonderful project that we're talking about today, and the team that I've introduced already. And so I just wanted to show you this wonderful video that the California Academy of Sciences made here for us to introduce the project. And then we'll go over some background with the help from our team. So hopefully you can see and hear this.
We're empowering our citizens, Californians, to get out there and say, hey, these are your marine protected areas. That's the coolest thing, is like, all you really need is your phone, and you can be a scientist. You can help us directly in Sacramento make better decisions.
When you take a photo and upload it to iNaturalist, you are making a record of a given organism being in a given place at a given time. And then with some clever AI and helped by the iNaturalist social network, iNaturalist actually helps you identify what you saw. We take your observation and put it together with the observations of thousands of other people and look for trends in the abundance of different species and the diversity of ecosystems that all this information combined can tell us about.
The state could really use this data to help us understand how our MPAs, our marine protected areas, are performing and how we can better manage them. But we need some help mining this really rich but messy data set for answers.
Right now, we're working together with the Ocean Protection Council, with our data scientist, Geo, to analyze all those data and help figure out how best those data can help inform marine protected area management and our understanding of how those areas are protecting biodiversity. Every year, we run a big campaign called the Snapshot Cal Coast, and that is people up and down the coast of California over 2 weeks in June, getting out to their local coast, the places that they love or the places that they know best, and making and sharing observations of all the species that we can find.
It's a pulse of information about coastal biodiversity, and it provides me with most of the data I need to understand the state of our coastal ecosystems.
So knowing what lives in MPAs helps us understand if our MPAs are working, right? So we need to know things like, are MPAs increasing density of certain species that are likely to benefit from MPAs? Are MPAs increasing biodiversity? Are MPAs maybe helping our ocean and coast be more resilient to climate change? So these are some pretty big unanswered scientific questions.
Because we've been monitoring for a while, we've been able to observe some big changes. If you see big trends, you can make some big decisions.
This project is getting us a sense of like the health of the coast and ocean statewide. It's a way to really involve people in marine protected area management and ocean management issues more generally, saying like, hey, get out to your local marine protected area and help make some observations that are going to help us make better decisions up here in Sacramento.
Even by making a single observation, you are providing the vital information that we need to understand what's happening to our coast and therefore influence the policy decisions that rely on that information.
All right, awesome. Hopefully you saw and heard that. So I just wanted to ask our dear friend Sarah Warden here, if you could join us.
course. Hi, Gio. Can everyone hear me?
Yeah.
Okay,
I can
great.
hear you. I can hear you.
Thanks so much for that introduction. My name's Sarah Warden. I'm an environmental scientist with the California Department of Fish and Wildlife, and I work on our MPA management project. We are the lead managing agency for the MPA network here in California. And just a brief overview, I'm sure most of us on the phone and the webinar are familiar with the statewide network. But just to give a high level overview, we have 124 MPAs here in California that are part of basically one of the largest scientifically connected networks in the world and the largest in the US. This network protects about 16% of our state waters. And as you can see by the pie chart, about 9% of the MPAs or actually 9% of those state waters are protected in state marine reserves, which are all no take. And there are several other types of designations included in the network. And they're primarily within state marine conservation areas that have some limited amount of take kind of based on the regional stakeholder needs and other things like that. So since the MPA network has been implemented, there has been a statewide massive effort to collect ecological and human dimensions data across the network. And we've of course partnered with people like Cal Academy, many of you on the phone, and colleagues up at Ocean Protection Council to undertake this monitoring program. So with that, I'm going to pass it off to my colleague, Mike Escrow from the Ocean Protection Council, and he's going to talk a little bit more about the monitoring program and how citizen science is getting integrated into that. So thank you for joining today, and it's been wonderful being a part of this project.
Great. Yeah, thank you, Sarah, and thanks, Gio. It's so exciting to be on this webinar today. This is really the culmination of a lot of work. So yeah, really happy to be here. My name is Mike Escrow. I'm the Marine Ecosystem Program Manager at the California Ocean Protection Council. And the Ocean Protection Council really serves as the governor's advisor on ocean policy. Our job is to kind of stand in between the research community and the decision-making community and ensure that the decisions that get made in Sacramento are based on the best available science and are based on our current understanding of what the most current data is telling us. We work hand in hand with the Department of Fish and Wildlife. We're being, it's OPC more on the management or the policy end and CDFW more on the management end. But both OPC and DFW have long supported the inclusion of citizen and community scientists in ocean and coastal monitoring efforts. And that's for a couple reasons. So as you heard in that video and a little bit from Gio in his opening remarks, citizen science can really supplement traditional monitoring programs. So you can have broader geographic scope, you can have broader temporal scope. Also, just as importantly, involvement in citizen science can really inspire stewardship. I think, a lot of you guys on the webinar right now are Snapshot Cal Coast volunteers. And I think participating in those kinds of events really brings home that sense of stewardship, that sense of ownership over our coast and ocean. So OPC's objective for this project was really to think about all of that crowdsourced citizen science data, all of that messy data, and really dig into it and see what kind of insights we could glean on MPA effectiveness, since this was really a project focused largely on marine protected areas, species distributions inside and outside of MPAs, but also, as Gio alluded to, broader metrics, right, that are related to biodiversity, representative of ocean health in general, especially related to climate change. So yeah, we're really just so excited with how this project turned out and excited for next steps, which Gio will talk a little bit about at the end. But I'll sign off for now and pass it back to Gio.
Thanks so much, Mike and Sarah. And actually, I'm going to pass it on to Rebecca.
Allison is going first. Actually, Allison is first.
Oh, Allison is first. Okay. So Allison Young, I'm having trouble moving through the slides here. Allison Young, co-director of the citizen science team here at the California Academy of Sciences, is going to tell us a little bit about citizen science and initiatives on the coast.
Yeah, thanks, Gio. So I'm sure most of you are familiar with what citizen science is, so I'm not going to bother going through that. But I wanted to talk a little bit about the goals of our program itself. And I don't know, Gio, if you want to, there you go, that slide, perfect. Yeah, so at the Citizen Science Department at the California Academy of Sciences, we're really focused on biodiversity documentation, kind of is the main thrust of all the programs that we run. The goals of our program, kind of overarching, are to connect people to their local nature, to really have people understand and appreciate and really embrace the fact that biodiversity is all around them all the time, no matter where you are. Then we want to connect people to each other. We really want to build community around nature. So almost all the programs we run have some sort of kind of community building aspect to them, where people can come together and meet other people who are interested in nature and learn from each other, and not just Rebecca and I. And then the science side of it is that we want to collect biodiversity data for science management and conservation. And so when we first started our program, Gio, you can move on to the next slide. We really wanted to have part of that biodiversity documentation and connection to nature have a coastal aspect to it. Both Rebecca and I are marine biologists, and we definitely believe that one of the best ways to connect people to kind of the oceans as a whole is to get people out to the coast, because that's the most accessible way for people to really kind of see the amazingness, the amazing biodiversity of our oceans. And so we started a citizen science program at the Pillar Point Reef, which is just north of Half Moon Bay. If you're not familiar with it, it's the same reef that forms the famous Mavericks waves for the Mavericks surf contest. And one of the reasons that we decided to focus particularly on Pillar Point Reef, not only just because we wanted to have people out at the tide pools and to do some citizen science out there, is that the Marine Protected Area Network had recently gone into effect, and just to the north of Pillar Point is the Montara State Marine Reserve, which includes the Fitzgerald Marine Reserve. If you've ever been out to that reef out there, it's pretty amazing. And although there's, it's totally adjacent to it, the actual reef at Pillar Point itself is not part of a marine protected area. And so it was designed basically to serve as a control to the Fitzgerald Marine Reserve and the Montara State Marine Reserve. And so when we decided to work out there, we realized there was not a lot of publicly available information about Pillar Point Reef, especially like not a species list or things like that. And so we really decided to step in there as a potential space where we could help at least provide some information about what's going on at that reef. And so we've been doing some long-term monitoring out there for over 8 years now. We started our program in 2012, where we collect data about a suite of species and some permanent plots that we have out there. But one thing that we always do with our volunteers is we spend some time after we've done our surveys to actually just search the reef and to document the species we find on iNaturalist to kind of help us build the species list at Pillar Point. And I think the slide is actually old enough because I know we have over 600 species that we've documented out there now, and made over 20,000 observations with us and our volunteers and other people who visited Pillar Point. So here's just kind of a nice overview of just not even close to those 600 species, but we have a really great idea now of the species that exist at Pillar Point and also when they're there. We have a great a sense of seasonality and when something new and unusual shows up as well. And so that was one of the great things that we realized about working at Pillar Point with volunteers, is that our volunteers not only were there to help us collect data, but they actually got to know that place really well also. And they are amazing at recognizing almost right away if something new or unusual is showing up and happening at that reef. So you can move on to the next slide. So one of the things we were able to document was sea star wasting disease. That was definitely something we were able to see out there. And then also the recovery from sea star wasting disease, that we started to see smaller stars and then the recovery of adult stars throughout time. And one of the other kind of big things that we saw out there with the last El Nino was this bloom of Okinia rosacea, the Hopkins rose, which is normally a very rare species at Pillar Point Reef. But quickly, by 2016, was the most abundant nudibranch out at that reef. And actually it was our volunteers who noticed one day when they were out there, the fact that they saw 3 in one day, they knew that was super unusual. And so they were actually one of the very first to kind of document this unusual occurrence happening because of our warmer waters due to El Nino. So we really saw the value of having people out at coastal places, and we knew a ton of information about Pillar Point itself. And we really wanted to think about how could we scale what we do at Pillar Point to other places on the coast. So with that, I'll turn it over to Rebecca to talk about the beginnings of Snapshot Cal Coast.
Great. Thanks so much, Allison. It's nice to take a trip back in time to our work at Pillar Point. So like Allison said, we knew that we were observing these changes that we'd heard about, and we had some data about those changes happening along the rest of the coast, but we really recognized the value in the volunteer corps that we've built who knew and were collecting data at one place. And we dreamed up, together with the Resources Legacy Fund, who are the initial supporters of this project, we dreamt up Snapshot Cal Coast, which is a way to mobilize volunteers up and down the California coast at one point in time. So every summer since 2016, over 1 to 2 weeks in June, we've been working together with volunteers to get out and document all of the species that they can see along the coast in their local places that they know well. And together, we can look at all of those data and see not just changes at one place like Pillar Point, but what's happening all along the coast. So our goals were kind of simple, were to document the biodiversity of the California coast in every coastal county, in every coastal place, from Del Norte to San Diego, using iNaturalist as the platform. And we were interested, and are still interested, in documenting changes in species ranges, patterns inside and outside of California MPAs, and to understand how volunteer collected data could be used for long-term MPA monitoring and ocean management. And we realized after the first couple years that these data could be used, potentially could be used as an early warning system. And we started to see the value, not just for the individual people that were making observations in their places and getting to see and explore their reefs that they love in really different ways. We recognized that these data together really could be used for management. And we were really lucky to have partners at the Ocean Protection Council, especially Cindy Dawson and Tova Handelman, who sat down with us, and we really partnered on thinking about how we had all these data. We had this great infrastructure of volunteers collecting these data year over year. But what we really needed was a data scientist to help us really dive deep into the data. And so they kind of teed up together with me and Allison. We all worked together to tee up this project and to envision kind of what the rest of the webinar and the work that Geo has done that you're going to hear about today. So I want to talk a little bit about how Snapshot Cal Coast actually works in practice. So next slide, Gio. So, I mean, the only way Snapshot Cal Coast works is through partnerships. And so we partner with organizations and individuals up and down the California coast. We work closely with the MPA Collaborative Network, and Calla Allison and her team that run the MPA Collaborative Network have been amazing partners. But one of the reasons we knew we could do this was that there was this existing network along the coast of people who worked in education and research and science and enforcement that already met regularly and were working in their counties to do work in marine protected areas, but also most of them worked in other places along the coast as well. So we have worked together with these partners and more every year. And what we've done is we've talked with all of these folks to give them the tools to use iNaturalist, to train their volunteers and their communities to use iNaturalist, to hold events called BioBlitzes where they can bring people together to one place at one time to document as many species as they can. We make stickers and promotional postcards, work with our science and management partners to develop a most wanted species list, and we distribute that. And so everyone can get their local communities out making observations in the ways that work best for them. So for some people, that might be a 100-person BioBlitz, but for other partners, that might mean a special tide pooling trip for their docents, or it might be a group of students from a marine lab going out together. Or it might be a California naturalist course taking their students out together. So we don't tell people exactly how they have to do it, but there's some basics that we help train our organizers to convey to their volunteers. So the individual volunteers and individual iNaturalist users who are making and sharing observations are really the engine, and we couldn't do any of the work that you're going to hear about today without all of these people working together with one common goal. So next slide, Gio. So the core really, what we have in past years, we'll talk a little bit more about how 2020, how this year is different, but really what we've done is encourage people to get out to one place. Like this is the Frenchman's Reef, which is just north of Pillar Point, where we do most of our work. And this is a little BioBlitz that we did during Snapshot CalCoast. So I kind of want to show you how this works. So next slide. So you just get a group of people out to the reef. Go ahead, Gio, that's fine. Get out to the reef, exploring and documenting and taking pictures and uploading them to iNaturalist. Next slide. So you can see this is just a group of 14 people out one morning, and together we made about 370 observations of 148 species just in one morning. And when Snapshot CalCoast is happening, this, next slide, Gio, is happening up and down the coast. So if you imagine this happening all at the same time in different ways, but all up and down the coast. And this is how we collect local data that's important to people about their beaches and their backyards, and then we string it all together to get a picture of what's happening across our entire coast. So you can see this year is the 5th year of Snapshot CalCoast. These are all of the data that we've collected together. You can see the first year in 2016, we made about just over 7,000 observations, and we were thrilled with this outcome. I think about 400 people made observations that year. But you can see how it's grown every year since. And so together, about over 4,000 people have made observations during the Snapshot CalCoast period that have helped us generate 71,000 observations of almost 4,000 species along our coast. And these data are the kind of backbone or the framework for the work that Gio is doing. And he'll explain a little bit more. He'll spend a lot more about that next. And we'll get a chance to talk a little bit more about Snapshot CalCoast 2020, but as you can imagine, we won't be doing in-person BioBlitzes in June. We're just gonna ask people to try and get out this summer and fall and make observations safely and legally at beaches near them. And we'll keep our fingers crossed that we can get together in 6 months in December to do a more traditional type Snapshot CalCoast. All right, that's it for me and for Snapshot CalCoast.
Thank you so much, Rebecca and Allison. That's an awesome overview of Snapshot CalCoast and the citizen science initiatives on the coast. And so I wanted to get back to thinking about the data a little bit. And in particular, one distinction, well, first of all, highlighting the fact that there are other amazing citizen science projects on the coast, and that some of those are much more tightly integrated already in the MPA monitoring efforts run by OPC and CDFW. And so those are, for instance, Reef Check and CCFRP and MPA Watch, and then the additional programs, Limpets and Beach Watch, run by the Greater Faroe Islands Association. And so in thinking about those, other than highlighting them as amazing programs, I just wanted to talk a little bit about why perhaps these are a little bit easier to intimately tie with other MPA monitoring efforts that perhaps run by professional scientists. And what it boils down to really is the fact that these volunteer-based efforts here in this slide actually follow a structured survey design. So a lot of them actually sort of use survey designs that are also used by professional scientists or have been co-developed by professional scientists. And they're just much more, there are much more tight guidelines around how the data are collected in these initiatives. And so compared to the data we're dealing with here in iNaturalist and the data that are collected in the context of Snapshot CalCoast, the real difference there is that there is a lack of structure. We call those unstructured crowdsourced citizen science data, and perhaps unstructured doesn't give them enough credit. Rather, what it is that there are a lot of different structures, combined in the same big database. So in that sense, it's very noisy because people collect data with many different purposes using different types of survey designs. So that's an important distinction to keep in mind. However, the lack of structure also means that you get an incredible amount of engagement, as we've heard. And so this here is this animation here shows the amount of data, iNaturalist observations on the coast. And these are research grade observations. So they're observations that have been not only collected, but have precise coordinates and timestamps associated with a figure. And the IDs have also been vetted by members of the iNaturalist community. And you can see the amazing peaks that we get in the summer ever since 2016. And that's Snapshot CalCoast together with the City Nature Challenge for those of you who know that other program. But those campaigns combined really do lead to a strong pulse in observation effort. So there's an amazing abundance of information here. So just to go over quickly the project objectives here, 1 and 2. The first one was to provide recommendations for trying to make sense of these noisy data and extract some clear and reliable biological signals that could be used for management of MPA biodiversity and coastal biodiversity in general. And the second one is to actually use those approaches for extracting biological signals and generate new knowledge of California coastal ecology and some of the things that are on our wish list and that have already been highlighted. And so, for example, the influence of changing ocean conditions and species range shifts and changes in population. So this brings us to this question, which is how can we extract signals of biodiversity change from these types of citizen science data, which are unstructured and crowdsourced? And those are challenges. On one hand, they enable us to receive huge amounts of information across space, time, and taxonomy, and that's amazing. But on the other hand, the lack of understanding of how those data were collected makes it challenging to analyze those data, and those data cannot be taken at face value. What exactly are the challenges that we're facing with those data? Well, there are 3 main types of biases that I want to talk about. And the first one is that there's an uneven coverage in time in terms of how many observations are taken and therefore how many species are documented in space and time. And that's because more and more people are using iNaturalist. More and more people are taking part in Snapshot CalCoast, and that's amazing. But what that means is that we have the curve here on the right shows the cumulative number of iNaturalist observers over the years, over the last decade. And as you can see, that's been going up exponentially. What it means is that in concert, observations of all major phyla on the coast have been going up exponentially as well. So if we were to look at face value at virtually any species or any taxon on the coast, it would just be going up. Though there is some variation in just how fast it is going up, and that has to do with how diverse the taxon is in question. The second bias, which I'm sure you're aware of, is an uneven coverage in space. And what that means is that there are places that receive a lot more activity from citizen scientists and iNaturalist users. And in fact, there are places that are better covered than others during Snapshot CalCoast, even though we try to compensate as well as possible there. And so the map here on the left shows you what the density of observations looks like. And we find that it's pretty tightly correlated with the number of coastal access locations across the coast, and specifically coastal access locations that have parking sites, because they make it exceptionally easy to be able to go down to the tide pools. So another bias in space is obviously that the number of observations in space there is due to the fact that observers are distributed unevenly across the coast as well. And so this is the pattern of iNaturalist observers on the left here. And you can see that that is pretty tightly correlated with human population density across the coast. So big cities generally have a higher number of iNaturalist observers, unsurprisingly. The third type of bias, which is actually very important as well, is that there is an uneven observation effort across space, time, or taxa. That is, there are some observation events, let's call them that, and we'll talk about these in more detail in a minute, but some observation events see a higher effort than others. And so, for instance, a BioBlitz during Snapshot CalCoast is a much more detailed sampling event on iNaturalist than perhaps a single observation during a hike in one morning unrelated to Snapshot CalCoast that maybe generates a couple of observations. And so in the map here, you can see the number of visits or observation events that have generated more than 10 observations across the coast. And those vary as well across the coast. So in the South Bay there, where we're particularly active, in Pillar Point, you can see a bright red spot there. On the right, there's a breakdown of the percentage of visits of these observation events every year that generate, that basically observe 1, 2, 3, or 4 or more species. And you can see that the vast majority of those generally involve the observation of a single species, perhaps with only a single observation. However, there are 10, 15% or so every year that involve the observation of 4 species or more. And so we have to be able to handle this diversity in, as I was saying, the structure of different types of iNaturalist observation events when we analyze those data. And so over the last 18 months or so, I've put together a recipe of sorts. I love cooking, so I turned it into a recipe that involves 5 steps to extract signals of biodiversity change from crowdsourced citizen science data derived from iNaturalist. And so we'll go over these in a little bit of detail with some findings here over the rest of this part of the webinar, because I want to give you some idea of the complexity that's involved here in analyzing data. So the first step, perhaps unsurprisingly, is that there does need to be some degree of filtering the observations. We can't use all observations, even though we'd like to use those, but we have to discard some of the information that is provided. And the first big filter is that in anything I have done and any of the outputs that you will see, we're looking at research grade observations. And so research grade observations are observations that have a photo associated with them. So an evidence of the occurrence of a given organism, and they have GPS coordinates and a timestamp associated with them. And those are really the basic metadata that we need for analyses. And especially the taxonomic identification is considered reliable to the point where these are the data fed to the Global Biodiversity Information Facility that we were talking about earlier. So the first big filter is we're looking at research grade observation. But filtering, it can be a lot more helpful than that. And for example, it can help to even out some of these those uneven instances of observer effort and observation effort across space, time, and taxa. One way, for example, that we've done that is in looking at change in ecological community composition over time. So what that involves here, you see we're providing this this visualization of ecological community composition change from 2014 to today for 5 different counties. And the way that we did this is not looking at face value, but we applied a number of filters. The first one is we're looking at research grade observations, but then what we use is this method of rarefaction, which is frequently used in ecology and, for example, in paleo data, fossil data. And what you do is subsample samples that have more observations than others. So find the minimum common denominator, as it were, in terms of the number of observations between different years in this case, and then subsample that number every year and look at the similarity in ecological community composition between those subsamples and not the entire sample. What that means is, for example, if I'm looking at 100 observations, I'm only comparing sets of 100 observations. Out of 100 observations, how many of the species were the same in 2 different samples? When we look at that across years, you can see, for example, that LA County saw huge dissimilarity in ecological community composition, a big spike in change compared to certainly the normal rate of change in 2015, which we'll see was a big year, and I'm sure you're aware, associated with the warm blood. The picture here is the same, but for coastal regions. And this shows here that the central region generally has seen a higher rate of ecological community composition change since 2013. And that makes sense from a global change point of view, because generally the central area between 2 extremes tends to see a lot more turnover from species moving northward as well as species moving southward, and therefore much bigger changes in the middle. The second step is actually to aggregate data across space, time, and taxa. And essentially what that is doing is making joint inferences or summaries based on many, many data points, whilst reducing the influence of any single data point on your conclusion. So that way you're hoping to dilute the effect of bias by trading off resolution across space, time, and taxa. For example, as you'll see in the tools that we'll have a look at later, we provide watershed scale results as well as other larger place-based results, and we don't go so fine in space because we're aware of the issues in doing so. But at the watershed scale, we're a lot more confident that we have complete species lists, that we can look at differences in the community composition between 2 different watersheds, and I'm much more confident in doing so. Another essential thing that aggregating data across space and time achieves is this idea of reverse engineering survey structure. So we were talking about how iNaturalist data lack an overall structure, and this can be done to some degree after the fact through data analysis. What that means here, what this graphic is showing here is just imagine the points, the black points here are just individuals of a species, and the blue triangles here are the observations, the iNaturalist observations of some of these individuals across a landscape. What we can do here is basically virtually throw some quadrats across this landscape and start combining some of these blue triangles together in an observation event. So I've been talking about observation events, and what that really is a footprint of a space, time, and taxonomy that allows you to combine together observations and think of those as one sampling event or one sampling visit. The way we've been defining these in the analyses that you'll see are events from a single user. So those are all observations from a single user in a single day within a spatial boundary. And at the finest resolution, the spatial boundary is around a 10-kilometer squared grid cell. So that is the basic analysis unit that we've been using. What does that allow us to do? What does combining some observations allow us to do? Well, it allows us to, for example, estimate how intense that sampling event was from a single user in a single place at a single time. And so these, for example, are two sampling events, visit A and visit B. And the first one is a relatively low intensity sampling event. So you can see on the y-axis there is how far the person walked between different observations. And on the x-axis is how much time it took how much time the person took between different observations. And so we have a total of 3 observations here in relatively short time, 25 minutes and about 100 meters or so. On the right here, visit B is a much more comprehensive sampling event where we actually have tens and tens of observations here over hundreds of minutes and walking across 1,000 meters or so. And so that looks like much more of a comprehensive sampling event on the coast. And we can start telling those two events apart and giving perhaps more confidence in the high intensity sampling one compared to the lower intensity one. This brings us to our second poll today, which is a little bit of fun, but I'm going to ask you what you think was the highest number of distinct intertidal species. So we're talking about tide pooling here, or on the shore at least. What was the highest number of distinct intertidal species observed during an observation event in 2019? So I'm going to launch the poll. Off you go. Distinct number of species. I'll give it a few more seconds here. All right, I'm going to end the polling. Most of you thought it was 74, which would be incredible, but it was actually 65, which is still absolutely incredible, given the numbers that we've seen already. 65 species during a single event is really a lot in 2019. And that is a lot for any type of sampling. I mean, 65 species, individual species, so those are not observations, but it could be out of hundreds of observations, is really an incredible sampling event and very rich, and something that we can use to tell pretty comprehensively what the community in a given place at a given time looks like. So well done to that person. I'm not gonna highlight who it was, but another crucial thing that aggregating different observations allows us to do is this idea of borrowing strength across taxa. What it means is that we don't actually want to look at just a single species, the observations for a single species when we're analyzing our naturalist data. And that's even if we're only interested in patterns for that specific species. That's because other species can tell us a lot about the sampling process of our focal species, the species that we're interested in. That's because they could be ecologically related to it. They could be, for example, prey or or a host. But also overall, they tell us about the sampling biases in the data. If we put together all the data, we're able to better understand things like uneven sampling over time, uneven sampling over space, and the types of biases that we've been talking about. So I'll give you an example of it. And here we're getting to some of the main indicators that we derived from our data. So if we look at Pysasta ocracia, a species that we all know very well on the coast, if we look at just the face value, the iNaturalist data, we see an increasing curve, like, as I was saying, pretty much anything else on the coast. But we know that that's not the case. What we have to do is look at observations of Pysasta ocracia as a function of observations of other species that had been collected either with it in the same places and over the same times and by the same users. And so those could be species that are ecologically related or other, for example, charismatic species on the rocky intertidal that tend to be recorded if Pysasta is observed. And that's the blue curve that you see here. Those are the other species that tend to be recorded together with Pysasta. And that's based on analyzing all the data and deriving clusters of different species that tend to be recorded together. When we do that, we look at the red curve as a function of the blue curve. What we get is this pattern here, which is the proportional rate of observation on iNaturalist of Pysasta ocracia, given that associated species were observed, or as a function of associated species. And you can see that this curve here looks a lot more like what we'd expect for Pysasta, with a huge decline from starting in 2013, but really in 2014, 2015, and then to do with sea star wasting disease, which we found out a little bit about earlier from Alison, and then a recovery in more recent years. How is this useful for management? We've talked about early warning indicators of change and this idea of an early warning system for the coast. If we look at patterns of proportional rate of observation, we can start seeing changes as they happen, formalized, not just by saying people have been seeing Ocenia more frequently, but actually we can formalize those in exactly how much more frequently than usual have people been seeing Ocenia. In this case, for example, we can look at the pattern with red abalone and other species of special interest. And you can see that abalone here, perhaps surprisingly, looks like it's increasing in the proportional rate of observation. And here the story is that red abalone is not doing well across the coast, as many of you might know, but it's doing extremely badly in the subtidal, which has meant that it basically is looking for food in the intertidal. And this means that people are seeing it more and more often in the intertidal, oftentimes unhealthy individuals, perhaps even dead individuals are being documented. And so we're seeing this pattern of increase in the intertidal, given to the fact that it is seeking refuge out there. This is Ocenia rosacea, the species that nudibranch we've been talking about. And this is a plot taken from our tool here, but you can see that the observation rate has been spiking in 2015, together with the warm blob, the episode of unusual warming of sea surface temperature, mainly in southern central California. And so that's the curve that you see in gray. It's the rescaled temperature maximum there. And you can see that Ocenia has followed that pattern almost perfectly there. However, I just wanted to caution against thinking that the warm blob was good for nudibranchs in general. This is the pattern across all nudibranchs statewide. So we've combined the reporting rate for different nudibranchs here, an idea of their population size. And we see that actually in 2015, 2016, the overall rate of observation of different nudibranchs went down, which means that generally the same nudibranchs were being observed, and there was a lesser diversity than usual. Another warm-loving species, perhaps, Velella velella, the by-the-wind sailor. There was a spike there as well, although the spike preceded, to some degree, the warming, and so it could actually be due to changes in wind strength or wind direction, bringing a lot of these individuals to shore. But there was certainly a significant increase in the observation rate of Velella velella individuals. So a lot of you are thinking, okay, well, just how reliable are these trends? And we've started doing some validation of these trends using the amazing data from the multi-agency Rocky Intertidal Network, or MARINE, and we'll talk about those data a little bit more. But those enable us to compare the trends we've been deriving from iNaturalist with more standardized long-term monitoring data, and a program that's absolutely fantastic. So thanks so much to the MARINE folks. So you can see that in some cases we get really pretty good matches. Once again, Pysastera craesus highlighted here on the south coast. We have almost an exact match there in terms of the declining picture. And then you can see some fluctuations are captured fairly well. However, in other cases, we do not get such good matches with marine data. And here, there are all sorts of reasons why we might not be getting good matches. And so we're trying to disentangle why that is, whether actually that's, an unreliability in terms of a population trend from iNaturalists, something going on in terms of the observation effort that's particular to this species or this region. But other times, there might be lags between the two patterns, and so differences in scale. There are also different spatial scales. So marine data are collected at specific sites along the coast, and these here are aggregated patterns across sites where you see those either statewide or for particular regions. And sometimes these aren't particularly significant fluctuations. And so really, if you looked at it more, if you squinted at it a little bit more, these are all flat lines. So we're trying to build an understanding of the validation here and why it is that the patterns we get from iNaturalists sometimes work and sometimes don't work so well. And we think that that is to do with a mix of observation variables, so how often the species are being observed and how many observations it has on iNaturalist, and ecological predictors. How abundant, how locally abundant is the species at a given site or in time? Those things all matter. And so for the purpose now, we're starting to detail a picture, but in general, there's a high uncertainty in the iNaturalist-based picture if the species you're looking at has a low mean population size in nature and has a low number of iNaturalist observations, and the variation that you'll see in the percentage, the variation in the reporting rate that we show you online, if that is low, then there's perhaps not the need to read too much into it. We'd like to model the bias process ultimately. And so we want to integrate some of these things we've been looking at, the variation in iNaturalist observations and iNaturalist observers. And so the way we can do that is using different models combined. And so we want to model the ecological process. We want to understand the influence of temperature and the trend over time. But to do that, we want to combine a model that actually includes some of these correlates as well. The number of observations, the number of species observed, and other measures of the effort. And so those, for example, are the time spent searching as well, or distance traveled, some of the things that we started looking at. Ultimately, we realized that for some biodiversity indicators, it makes no real sense to look at iNaturalist data in isolation of other amazing data like marine, But really, if we want to get to the true ecological signal of biodiversity, we actually want to combine these things and combine their different strengths, combine the standardization of marine data with the broader scale of iNaturalist data. And statistical frameworks have recently emerged that allow us to do just that, combine marine data together with physical oceanographic parameters and the broad presence-only data from iNaturalist, put all of those in a model, and through joint inference allow us to get the types of conclusions that we'll look at in the live demo and the tools that we're providing. So just for some quick examples, for example, we've been able to look at the influence that temperature has on the distribution of Tetraclida rubescens, the pink volcano barnacle. And you can see that generally Tetraclida tends to increase in density at slightly warmer temperatures. Unfortunately, this is truncated here at the border with Mexico. And so, we're getting an upward curve, but obviously this should go down at some point when it gets too warm. But certainly in California, the warmer it is, generally, the more this species increases in density. And we've been able to see how this has led to northward shift in the density of the species over time since 2014. Another species is the owl limpet. I won't go too much into detail here. We'll have a look at it live in the tool, but it's another species that's shown increases in density due to its warm loving. And so once again, these are results that combine marine multi-agency rock intertidal network data together with iNaturalist and physical oceanographic parameters. And that's really what we consider the gold standard ultimately for integrating iNaturalist data and crowdsourced citizen science data in monitoring efforts here on the California coast and beyond. So I'm going to leave you there for 7 minutes or so. We'll be back at 3:50 for a look at the outputs that we put together here and show you how you can access the results that we've been talking about. So thanks so much, and I'll see you back here just in a few minutes. And please line up your questions in the Q&A here. We'll hope to have a live Q&A, but you can also help us by including your questions here. If you can find that there's a Q&A button on your screen where you look at all the menus there, and you can include your questions. So that'll be wonderful. Thank you very much, and I'll see you in just a few minutes. Hi everyone, we're back for part 2 of monitoring California's coastal biodiversity with citizen science. Sorry, I was just reading a Q&A question there. Thanks so much for asking some questions. Please carry on asking some questions using the Q&A, and then we'll open it up for discussion at the end. But the second part is really about talking about how we've been putting together outputs for this project, how we've been constructing our outputs and developing our outputs, and then how you can now access those online and in other ways for your own benefit for your work. So the first thing that I wanted to do is when you registered here, if you're an attendee of the webinar, we asked you to self-identify in a stakeholder group. And this is the breakdown of what you all responded. And so the first thing I wanted to highlight is that, you know, we're doing reasonably well in reaching different stakeholder groups, but we're not doing quite well enough. And there are some groups that that we know are underrepresented in our community, and the Snapshot CalCoast community, and generally in this type of work. And so we'd like to do better, and we want to continue to collaborate to do that. And in particular, for example, we started collaborating with tribal representatives in helping us identify how these tools and these types of output, if in any way, can be useful to tribes, and try to collaborate more and tie this work with emerging tribal MPA monitoring efforts that are led and developed by tribes. And one way, for example, is, as you'll see in the Places app, we explore things in different places, watersheds, MPAs, and we'd like to be able to have a tribal perspective there, and maybe include something that is akin to recognized tribal lands, or, you know, recognized by tribes. And the other group that you don't see here is really commercial fishers. And there, you know, there's been some effort with Snapshot CalCoast to try and reach out a little bit more, and really try and find out how commercial fishers can benefit from this work. And so we look forward to continuing there. We do have, however, a pretty broad representation of stakeholder groups here. And so that is thrilling. You're coming from many different groups. And so we're looking forward to continuing to expand our community here, and to hopefully make our outputs and findings and initiatives as relevant to as many of you as possible. So you're here today, which is wonderful. And we've been trying to engage with you and the community on the California coast that cares about California coastal biodiversity from different perspectives since the start of this project. One of the things we did is send out a survey very, very broadly to get ideas and thoughts on what people from different stakeholder groups might want to see out of this work. What kind of findings, what kind of species, what kind of areas are people interested in? What do they want to know? And so we asked that. We had an amazing meeting in person here at the Academy. We were going to have a second one, but we were unable to. But we had a really incredible meeting where some of you online, I hope you're online, you joined us here in person, gave us wonderful inputs, wonderful feedback. And I want to highlight a really crucial contribution that was that of Adrian Dahoud Fritz, who was there with us and really, I think personally, gave us a lot and gave me a lot to think about in terms of carefully crafting these outputs and thinking really deeply. And so I want to thank her and remember her for her amazing input and her, this project really did benefit a lot from her input and her help and her friendship. So we received a lot of great feedback during that stakeholder meeting last year, and we put that together in a report where we sort of summarized everyone's input. And this was sort of there by my desk as I was working on this over the last 18 months. And I would sort of periodically go back and look at different things that could be addressed in the tools and outputs that are coming out of this project. And the other thing that we asked people to do was to basically dream up their tool, draw their dream tool. And so I've got these drawings also near my desk. I scanned a few of them to show them to you, but people really gave me some wonderful ideas of how to put together a tool that would allow people from different backgrounds and different stakeholder groups to explore these data. And so, so these are two of them. And we have another two here. This one here on the left, I can highlight it as Mike Esgroves, I'm pretty sure. And it looks great. But I just want to say, first of all, thank you so much if you contributed your drawings, if you were here helping us last year in June. It was really, really useful and really amazing for me. And I wouldn't do any other project of this sort without doing something like that. And so I hope that the tools that we'll have a look at now sort of fulfill your sort of ideas and thoughts. And maybe they don't look quite as good as these, but I tried my hardest. So what we're gonna do, I think. Actually, I'm going to ask my my co-hosts here if they wanted to add anything in terms of engaging stakeholders and partnerships. I may have forgotten something. So just if you want to add anything, perhaps not. We can also revisit things during our Q&A. Rebecca, did you unmute yourself? No, sorry. Let's talk a little bit more about this during the Q&A, but I just wanted to take the remainder of our time here to introduce the web tools that you can use to access the findings from our project. And so what you want to do, if you do want to access the data, I hope you do, is navigate on calcoast.dob.bio, and DOB stands for the Dynamic Observatory of Biodiversity, which is the name that we've wanted to give here to these tools, because we believe that the real strength is in the fact that they're so dynamic and they allow us to gather data rapidly, almost in real time. The dream would be to make these real-time tools eventually. But what we ended up doing is dividing this up in two different tools. that allow you to explore the different types of indicators in different ways. The first one is an Explore Places tool that is a sort of place-based map here that allows you to look at different types of spatial boundaries and place-based biodiversity indicators. And so we'll have a look at this live in a minute, but there are 5 main place-based biodiversity indicators that we're deriving from iNaturalist data. In this case, this is exclusively iNaturalist data. So those are richness, uniqueness, rarity, irreplaceability, and change. And those words all apply to biodiversity. So we'll have a look at this in a minute. But the second tool is a species-based tool. The Explore Species tool allows you to look at specific species that you might be interested in. You won't find all of the species, as we'll go over in a minute, but there is a subset of species there that satisfy the filters and all the statistical things that we've been talking about in the previous part of this webinar. This is the Explore Species tool. If you're interested in a particular species and would like to know, for example, population trends over time or where to find it along the coast. So that's not a slide that I wanted to show you right now. So I'm gonna exit the presentation here really quickly and take us to calcoast.dob.bo. So if you navigate to that address, you'll end up on this landing page right here. Let me just quickly check the chat, make sure. Okay, sorry, there was potentially some formatting issues. Hopefully those are resolved. Do let me know if you can't see this, but hopefully we're all together on, all good, perfect. We're all together on the Calcoast DOB app. So the Dynamic Observatory of Biodiversity. So this is what the landing page looks like. And you can navigate to an about page here that just gives you some broad background of what we've been talking about. And in fact, you can find the video that we played at the beginning of all of this right here, and my contact if you want to get in touch about any aspect of this, or in fact, any aspect of what we've been talking about today. The other thing that I want to highlight is there's this methodology tab here that will open a new window that will help you understand the methods in more detail. So if you're interested in finding out the definitions of the different places that we've used, or if you're interested in finding out more about the data, iNaturalist, marine, or some of the oceanographic data and habitat data, or if you just want more detailed definitions and methods about the different indicators, I will advise you to come here and check all of that out. This is a little bit of a work in progress, so this documentation will become more and more rich over time, but right now you should have enough to get a good and pretty specific understanding of everything that's going on. So we're back on the landing page, and we're going to start having a look at one of the tools. So if you click Explore Places, you will be taken to the Explore Places tool, which looks like this. It's a map-based interface that shows you the California coast. By default, you're getting a watershed view here with the coastal watersheds. This is not specific choice by any means, but we certainly think that watershed scale results are informative. And as you'll see, watersheds satisfy a lot of the filters that we've been talking about. And so perhaps it is a little bit more rich. But you can switch between those. And for example, look at marine protected areas or counties as well, or hexagons. And hexagons are an attempt, these are completely arbitrary hexagons that are an attempt to get higher resolution picture of some of the place-based indicators that we'll have a look at. So today, we can have a look at MPAs, for example. And I'm not going to go in detail into what the different indicators are, but you know, species richness is fairly self-explanatory. It's an estimate of the number of species that are there. But I'll show you a little bit how to navigate this. So one way, there are really two sort of workflows that you can take in the Places app. And the first one is if you want to look at the network as a whole, you can add things to the map and then sort of navigate this map and basically look at how low or high the estimated richness is here between different places. And you can switch between different place-based indicators. For example, if we want to look at rarity, which is a measure of how many rare species are estimated to be in a given place. Note here that some of the indicators are divided, further subdivided in taxa. By default, the result is based on all species, all intertidal species. And if you want more details, once again, you can join the methodology tab from within the app. Not all of them allow you to break it down by taxa. So we can have a look at sort of the network as a whole. If you're interested in comparing different places or looking at variation across the whole coast. This button right here, the clear map button, is useful. If you're stuck or something weird is happening, you can clear the map and it will sort of reset everything and send you to the beginning. Another workflow, for example, if you're really interested in a specific place, is to just click on it. And you can do this at any point. So let's say we're interested in actually Pillar Point, right? We've been talking about Pillar Point. And so what this allows you to do is have a look in more detail at what is going on at Pillar Point. So for example, we can look at change over time. Sorry, the boundary is such that all the Pillar Point results are actually in here. Let's look at Montara. So this here, the ecological community change measure is what we talked about a little bit in the previous part. And that's the way that composition has been changing over time, year on year, in this particular place. And it gives you an idea of how much change was happening in the ecosystem at that time, based on all species combined. A handy thing if you're interested in a particular place is this report feature here, which allows you to get all the measures together for a given place. And so you see here that for Montara, we're getting estimates of species richness, biodiversity rarity. These are expressed as percentages, and really the percentage reflects how highly it is ranked compared to other MPAs. So for example, in terms of irreplaceability here, which is defined in how key that MPA is for, you know, in theory, if we were to safeguard particularly rare species and species that are really unique to this MPA, that gives you a higher irreplaceability score. And so you can find that Montara is relatively high biodiversity irreplaceability. And then we see the ecological community change picture here. Once again, interestingly, and perhaps unsurprisingly, 2015 saw higher change than usual. 0 here is the background rate of change. So hopefully this gave you an overview of the place tool. We can, I'm happy to answer some questions in the Q&A, or feel free to email me and contact me if anything comes up. We're hoping to, as I'll talk about in the next steps, to add a little bit more complexity here, and certainly the opportunity to download any of these outputs and increase the content of the methodology here, so you can understand even more about what's going on in the background. But let's go over to the Explore Species tab, which is a different tool. It allows you to have a look at species specifically. And so you have 3 indicators here: the population trend, which shows you the proportional rate of observation of iNaturalist, which is that quantity that is sort of akin, gives you an idea of population fluctuations over time, and we had a look at in the previous part of the webinar. And here, for example, so you can either scroll through these, or by hitting backspace, you can either search for something specifically. So let's say, for example, we're interested in the ochre sea star. We're getting the results here that we had a look at before. And here on the right, you get a breakdown by region. And the reason there isn't a mode, and these are the 3 regions here and how they're defined, which is the Ocean Protection Council's definition. But the reason you don't get a more detailed spatial breakdown here is precisely because the data are not rich enough to allow a more detailed spatial breakdown that still has the same resolution over time. So if you remember in the previous part, we talked about trading off the scale of different dimensions. And oftentimes you have to trade off spatial scale with temporal scale. So if you want yearly trends in a particular species and want those to be somewhat reliable, you need a decent quantity of data. And if you go too detailed in space, generally there aren't enough data as of yet on iNaturalist to be able to get enough detail. Let's have a look at Oceania, for example. So one feature here that is useful is that this allows you to actually go and visit and have a look at the underlying iNaturalist observations. So these are the last 6 observations of Oceania on the California coast. And you can click on one of those, and this will take you to iNaturalist and have a look at exactly what this observation was and where it was taken, and then just navigate the beautiful interface that is iNaturalist if you want to find out more about Oceania specifically and all the functionality that iNaturalist provides. Another useful thing here is that you can actually add a physical variable here if you want to have a look at what the fluctuation in that variable was together concurrently with the fluctuation in your population. So we talked about Oceania and how its population sort of went up significantly at the same time as the warm blob. And so we can sort of start testing that hypothesis by plotting, for example, maximum temperature on that curve. And this is the figure that we saw in the presentation earlier. And here temperature is unitless. It has been rescaled between minimum and maximum to show you the pattern of fluctuation here, so that you can compare them directly. So let's have a look at some of the other indicators that you can use here. And the first thing to know here in the next two tabs, geographic range and range change, is that you have a lot fewer species. And these species, if you remember from the presentation, these two results, geographic range and range change, actually come from a model, an integrative model that combines iNaturalist data together with marine data, the multi-agency rocky intertidal network, as well as oceanographic data, which you can already see here, that come from UC Santa Cruz's ROMS model outputs. So here we can have a look at the owl limpet, for example, which we didn't have a time to look at earlier. And the geographic range, make sure you hit show range here after selecting it. There's two basic outputs here. The first one is a map of density over 10 kilometers squared grid cells here across the coast that shows you, this is the model output. So this is not the frequency of iNaturalist observations. This is the model estimated density based on jointly the marine data, iNaturalist data, and physical oceanographic factors and habitat data, in fact. So this is as close an estimate as we can get to the density of the species on the coast. And you can see that here we have a different output from the model that actually shows you how population density on the y-axis changes with some of the physical parameters that are included in the model. In this case, temperature minimum, but you can also look at median salinity, bathymetry, and maximum sea level. So for example, we can look at sea level and see that the owl limpet wouldn't do so well in terms of the estimated density here is lower as maximum sea level rises. Perhaps I'm not making the conclusion because this is not a forecast, but perhaps sea level rise wouldn't be such a good thing for it. So the list of species here is the list of species that satisfies the data availability requirements. So the species that are included in the marine long-term plot or count surveys, and that have enough data on iNaturalist, and that are generally recorded at the same time on iNaturalist as well to address some of the bias issues. The range change list of species is the same, but range change actually is the output of a slightly different model that includes a year component here. So the distribution is broken down by year, and it allows you to have a look at the change between different years in that in the estimated density of that species. So you can see that between 2012 and 2019, there was some estimated increases, and these are sort of substantial increases in the estimated density proportionally. So there are some estimated increases in the density of the owl limpet in the here the Monterey Peninsula, the Monterey Bay, and Southern California here. And you can play around with the years here to find out some differences. So let me check the time, make sure that we're on top of it. I think I'm going to have a look at the Q&A really quickly to see if there are any relevant questions to the tools. Just so that we can address these here. So we have specific questions about the temporal resolution. So the resolutions in terms of spatial and temporal resolution that we're presenting in these outputs are what we feel comfortable provides the most reliable pictures of any of the biodiversity indicators at this given scale. Any higher resolution in space or in time, if we were to look at, for example, monthly changes here, we're not confident that there are quite enough data at this time on iNaturalist to be able to reliably get a picture of fluctuation between different months. But this is not to say that this could change very quickly, or for example, we could do so just for the last couple of years. And so those are definitely questions to be addressed. But generally, to bear in mind is the fact that in analyzing those data, the trade-offs are there to make sure that enough data are actually included to extract reliable patterns. So I think this about wraps it as an overview of the tools. So I'll remind you that if you're concerned about any aspect of what you see in here in terms of the data that went in it, in terms of questions about resolution, or just what the definition of an indicator is, you can see, actually, I forgot to point you in the direction of this, which is a short description of what the indicator is. But if you want a more detailed one, you can just fly to species-based indicators here, and you'll find the different steps into extracting those patterns, and in fact links to the wonderful data sources that that we've used, as well as iNaturalist. So I'm going to end the live demo here and go back to the presentation. But please feel free to include your questions in the Q&A. Okay. Oops, this went all the way back to the beginning. So let me select, sorry about that. Let me select the last couple of slides. Oh no. Okay, I have to do it manually. Unfortunately, sorry about that. Close your eyes. Close your eyes. We're going through the entire presentation again. But I feel like this is the quickest way we're going to get there. Sorry about that. I can't seem to figure it out. Okay. Our wonderful drawings here. You can see that we're not too far. We're not too far from the drawings. Okay, so I just wanted to take the last part of this webinar to talk a little bit about the next steps for this project, and for, in fact, our partnership between the California Academy of Sciences, the Ocean Protection Council, and the California Department of Fish and Wildlife. and then we'll open it up for a Q&A. I'll answer some of the questions. And so the next steps immediately for this project, I've touched on some of those, are that we want to continue to disseminate the outputs. So today we've released the DOB CalCOAST website. So feel free to go on there, play around with the tools that I've just shown you, and just email me if any issues come up. I don't want to give the impression that this is not still a work in progress, and that things can't be improved and functionality added. So please feel free to email me, contact me if you have any questions. The other output that we haven't had time to talk about, but that will be released fairly soon, is a report to OPC and CDFW that details everything that happened in the last 18 months. So that's a lot of what we covered today, the background, as well as the specifics of how to analyze data and some of the findings, and then next steps and what to do moving forward. And then the last step is really to write academic papers. At least a couple should come out of this work to actually, because a lot of work here we think is pretty novel in terms of the analysis of crowdsourced citizen science data. And so we think it is a broad appeal to the broader academic community who might want to use iNaturalist data for different purposes or in different places. As part of all of this, we are committed to publishing and providing all derived products and software and code, in fact, to build the tools and generate the outputs as open source. So I'll be working on this to make sure that soon enough you are able to download the maps of different indicators and the figures that you see, as well as actually pretty much build the tool yourself if you wish to do so, maybe for a different place. And then obviously longer term, we're hoping to maintain these tools online and update them regularly as information comes through. So we talked about the dream of real-time integration here, real-time updating. It probably isn't going to be real-time, but certainly soon enough we'll want to integrate the snapshot CalCOAST 2020 data and hopefully more frequently than once a year for sure, but regularly update the outputs. And then, as I mentioned, increase the usefulness of the outputs for different stakeholders. We talked about, for example, adding different types of places in the Places app. For example, tribal lands is a real priority. It would be fantastic to include those, and we look forward to continuing those discussions. And other species of special interest that might not be included at the time. So please email me if you have any thoughts of something that would be particularly beneficial to you. If it looks like it might be particularly beneficial for many of you, then it's something that we can certainly consider including. And then another idea is that maybe these tools aren't the best to reach everyone. And so we are open to communicating results differently if that's a better way to reach other people. So I want to talk a little bit about the next steps for this partnership. And those are really to continue to move towards a better integration of crowdsourced citizen science data, engagement data and insights into coastal monitoring and management in California. And that's through these tools, but through additional programs and projects that we think can be beneficial, and we hope to start together. And for example, we recently discussed how Snapshot CalCOAST 2020 and in general Snapshot CalCOAST and iNaturalist activity can help compensate for canceled field seasons. This year, as you know, a lot of field work has been canceled to comply with shelter-in-place regulations, rightly so. But perhaps there's a way that, single INAT users by themselves with some guidance can help compensate for gaps in certain places and for some species that we might not be able to observe or find otherwise. And in it sort of continuing in that thought, with that thought, we're working on with OPC and CDFW on developing and formalizing an early warning and forecasting system for biodiversity change on the California coast. And that's really honing in, that's compared to the current tools, is doing two things. It's really honing in on the predictive forecasting potential of these data and these tools. And then really think deeply about vulnerability of places and species to environmental change. We think these are two real strengths of the Snapshot CalCoast community and iNaturalist data, is just really how rapidly they can provide information compared to other data sources and how broadly they can do so, the area they can cover. So we want to formalize this early warning system and put it in a predictive framework as to be able to model not just recent, but also projected future trends for species and places, and identify sentinel species and places that are likely to be subject to abrupt changes, and really provide guidance and engage people to sort of adopt these species and places as their own and really help track them more specifically and effectively. So with that, I wanted to remind you, this is why the slide is here. We haven't gone back to the beginning. I wanted to remind you that Snapshot CalCoast 2020 is happening this year. And maybe Rebecca, you want to jump back in here and let people know what's going on this year, remind them.
Sure. First of all, thanks, Gio. It was great. I'm looking forward to answering all the questions and helping curate it. But I first wanted to just say, awesome job of explaining some pretty complicated stuff in a super, super accessible way. So thank you. Because none of the questions are about like, I didn't understand what you're talking about. It's awesome. So I think it was great. And so Snapshot CalCoast 2020, this year for the summer and fall, instead of asking people to, go out over just 2 weeks in June, because many beaches, of course, still have restrictions, or some beaches are still closed and many of them have parking restrictions. We are going to kind of extend the summer recording season, or the recording snapshot time, to the summer and fall, basically like June 1st through the end of November. And our ask is actually kind of simple, but very different. So we're asking people who can safely and legally get to places on the coast to go out and make observations as many times as you can from those places. Ideally, it'd be great if people can kind of adopt a spot that is easy for you to get to, or easy-ish to get to, and go there repeatedly and document as many species as you can. We have a list of target species that will help guide and focus you a bit. And we also have a list of places that Gio has built by looking at the marine sites and places where we have a lot of iNaturalist data, or places that are pretty gappy for iNaturalist data, to help us build a picture together and fill in the spots that we're not able to monitor right now. So the communications about the summer fall Snapshot CalCoast, I'll be sending to all the participants and organizers and the community writ large in the next week. So we'll be on the lookout for that. Of course, we won't have printed materials like we normally do. It will be all digital. And we think this is a really important way that we can give folks, first of all, who are missing going to the coast, hopefully between now and November, people can get out there at least a couple times. So give us the connection to the coast that we all love, but also fill in some really important data gaps, because we can go alone or with our family units and make observations in a way that we professional scientists can't right now, because there are groups of people, or it's really, really difficult. So we can fill in some really important gaps. And fingers crossed, in December, so 6 months from the normal Snapshot CalCoast, we'll be able to do more event-based, albeit I'm sure smaller, but maybe we'll be able to do some event-based data collection for a kind of winter Snapshot CalCoast. And we've long wanted to have Snapshot CalCoast twice a year. And some of our partners like Crystal Cove in Orange County, they do it twice a year. So this might be a nice way to get in a rhythm of having a 2 times a year data collection going forward, and having people adopt places to collect regular data, so more places can have the kind of richness of data that we've been able to gather at Pillar Point, because people are going there really, really regularly. That's kind of the ideal for filling in some of these gaps that Gio described in his tool. So I'm happy to answer more questions about that in the Q&A, but I want to make sure that we have a chance to answer everybody's questions and have a richer discussion. So thanks so much. So Gio, I think, do you have another, do you want to go straight into questions, or do you have something else before we move to questions?
I just have one more slide.
Awesome.
And that's the Q&A slide, but most importantly, I think, is the overall conclusion of this webinar. And thank you so much for sticking with us this whole time. But I hope to have shown through this project really that crowdsourced citizen science data are a crucial tool to complement biodiversity monitoring efforts on the California coast and beyond, we think. But I hope that after this project, we'll be able to better think about how to integrate these data in biodiversity monitoring efforts, because there is a real richness here that really cannot be found using other data sources. And I think it would be a shame to disregard it or not considering it, because we might be missing something there, and that would be a shame. So hopefully I've gotten that point across, and will continue to make that point. But with that, I'm just gonna open it up to the Q&A. And the way I was gonna hope to do this and start was really to just start in answering some of the questions that have been asked in the Q&A window here. And then if there's some time, we can open it up live and give people a chance to speak, or really, or you could write some more questions as I go along. So please do use the Q&A tool if you find it. I'm not going to go over the chat too much because I can't go back and forth. But so the first question, which has 3 upvotes, is that it would be great to hear from OPC about how they're applying these data to MPA management decisions. And there is another one that I saw that was sort of similar. And that's, can you speak to the importance of Snapshot CalCoast? Wait, sorry, that's not that one. There's a question about how these kinds of tools can really help management decisions, and what are the policy levers that these tools can help address. And so I just want to maybe cover OPC a minute, just saying that this is a little bit premature in terms of already having helped OPC to make management decisions, because we're releasing the tools today and we're just getting to the end of this project right now. But maybe Mike, if you could speak to some of the policy and management decisions that could be affected by these data and tools, that would be fantastic.
Hey, Gio, this is Sarah.
Sarah.
I'd be happy to take on the management decision perspective of it from from CDFW's perspective. So I think you gave some great examples on your following slides, and thanks for pointing it out. Like, the short of it is OPC and CDFW are anxiously awaiting kind of GEO's final report out on all of this. At that point, we'll really be able to take these results, dig deep, and kind of start to roll out the types of management decisions that we'll really be able to apply this project to. But looking at it kind of from a higher level, just the ability for the iNaturalist data to kind of start detecting changes in communities across a broad region of the coast will certainly help inform kind of where to prioritize monitoring projects and species to look at, kind of drilling down even further than what the action plan has given us. And then, you know, one of the really kind of big important things from the department's perspective is that the iNaturalist data and the stuff that GEO's been working on really helps get us community buy-in to the MPA network and what we're doing from a monitoring perspective. And community buy-in and like stakeholder gives our stakeholders a voice in those types of management decisions that we are making, kind of no matter what they are. So we really look at these citizen science projects as being really important for that perspective too, just managing the network with all of our partners. And I will just take this opportunity to say, and kind of plug what's going on with OPC in the department right now, is that the department is required to update the Fish and Game Commission on a decadal cycle about the performance of the MPA network and kind of how our management program is doing, and that will be happening in 2022. So we will be rolling out a lot of communications with partners, meaning pretty much everybody on this call, about how that's going to go. And that's when we're really going to have a lot of concrete answers about things like the work that GEO is doing and concrete management recommendations to move forward. So short story, stay tuned, and then I can pass it on to OPC to add to that.
Thanks, Sarah. Thanks so much.
Yeah, thanks for that, Sarah. Yeah, we were definitely, you know, we're aligned on this. I was going to highlight the same things, the 2022 management review. You know, yeah, since OPC and DFW really work in lockstep, we're thinking about the same kinds of questions. So CDFW, you know, works a lot in those pillars of MPA management, kind of boots on the ground. You know, in terms of OPC, we're as sort of the other side of that coin, the policy lead on MPAs. You know, we're really interested in these same questions that really boil down to, you know, are our MPAs working? So, you know, what we do as OPC staff, we staff high-level decision makers. You know, our 7-member council includes the Secretary for Natural Resources, the Secretary of Cal EPA, the Lieutenant Governor. So these are folks who really, you know, all they want to know is, okay, how are these things doing? And we can glean insights from citizen and community science that that can help answer that question that we can't necessarily glean from more traditional monitoring programs. So in 2022, when the state is on the hook to really answer that question of how has this, you know, really globally significant network that we've built and spent so much money on and so much time and what feels like blood, sweat, and tears, right? Like, what has that actually gotten us? This effort will really help us build a much stronger scientific backstop to be able to answer the questions that those high-level decision makers in the policy realm have.
Awesome. Thanks so much for that. And yeah, I'm hoping I'll be able to continue to help you define all of that and formalize exactly how these data can help with that. Definitely an ongoing process, and you know, it's a complex question for sure. Okay, so there were a couple of people asked if there were efforts to collect data inland and whether these data might come into basically being integrated into the tools that we talked about. And so I'll just give a quick answer, but then Allison or Rebecca, feel free to step in if you want to talk about it a little bit more. So the first answer is that iNaturalist data and the iNaturalist sort of project has no spatial limits. So it's a global initiative, and so we definitely have data inland on the California coast. We made a decision. If you go have a look at the methodology of the whole framework, we made a decision to have a coastal boundary that's defined by basically coastal habitats in California. But that was to be the most useful as possible to this project, to OPC, CDFW, and MPA management, and to be attuned with Snapshot CalCoast. But there are data on the other side of the boundary, no doubt. And I will also mention that estuarine areas are included in the boundary. And so when you look at watershed scale results, those include coastal habitat in the way with the coast, which also includes estuaries and bays. So yeah, I think, I don't know if Rebecca or Allison, you want to say something about inland initiatives?
Sure, I'm happy to start out, and then Allison, if you want to join in. But the work that Gio talked about today is our coastal work, but within our team and our focus of the work that we do, we work all over California, and we also have a project called City Nature Challenge that collects data from city and metro areas from all over the world, terrestrial and along the coast. So really, we see this work that we've done through Snapshot CalCoast, as near and dear to our hearts because we love these places, but as a model for what we can do in inland California next, really, and then to build a set of tools that Gio's building that people could implement anywhere in the world. And as you can see from what we presented today, and it's a kind of two-sided set of tools. Like one is a campaign and community building and training and network building to collect data and to engage volunteers and continue to engage and volunteers and build a community of iNaturalist users. And the other is analyzing those data and modeling those data for all the biases as Gio described, and making them more meaningful for management and for making decisions. So we see this as like kind of the first place that we're able to do this, but we are super excited about scaling this work, especially throughout California, but to make those tools available for anyone, any place in the world. Allison, I don't know if you wanted to add.
I think you got it. Yes, we can make observations inland. Please do.
Yes. And I see Ariel's question, that's really about these like estuaries and coastal prairies and terraces. And, right now the area that we consider for Snapshot CalCoast includes most of those areas. If you actually look at the place that we use, I'm happy to share that geography with you. When Gio came on board, he helped us kind of standardize our geography, because before we had someone who was Gio with GIS skills or crack GIS skills, we had kind of a cobbled together place. And so we have a more uniform place now that we use for Snapshot CalCoast. And when I say place, that means the geography that iNaturalist is using to pull the observations that we're using. So many of these habitats that Ariel's described are included there. But our focus in data collection and campaign has always been the rocky intertidal. And because the marine monitoring is a robust, like shared protocol along the entire coast, which is actually very, very rare for one habitat to have one monitoring scheme that the team uses. As we find and move more inland with this expertise, we need to identify, this like robust community ecology monitoring that is across habitats to help do some of the modeling that Gio presented today. But all observations from the coast are captured in Snapshot Cal Coast. And if there's a place that has not been captured, we're happy to talk with you about including that.
Thanks so much. Thanks so much, Rebecca. And so I'm going to take this first question that was upvoted here, which is about how good the alignment is between species of interest to management, such as those included in the monitoring plan, and the species that INAT observers document. And thanks so much. That's an interesting question. And there are definite complexities about what INAT observers document and choose to document. And for example, as we've started validating data using long-term monitoring programs, which really focus on those species of interest more so, we've started seeing that generally more common, so that's more locally abundant species in the intertidal are generally pretty well documented by INAT observers. But there are two types of rare species, and rare species could be aligned with interest to management, or they could just be locally rare but not necessarily threatened or of interest otherwise. But in terms of rare species, there are rare species that are really well recorded on INAT, and then there are rare species that are not very well recorded on INAT. And the difference seems to really to do with how easy they are to find and how perhaps how charismatic or how much people want to find them. And so there is a set of species of special interest to management that are being missed a little bit by INAT, and that's the sort of rarer, harder to find species that are of interest to management. Those that are much better covered by standardized monitoring efforts. But overall, this really gets at the idea of what INAT data are perhaps best suited for. And we think that actually they are not a replacement for long-term monitoring efforts. As we've tried to outline here, it's really a complement to long-term monitoring efforts. But actually, they are amazing at detecting those species that might not be the rarest or most common, but or immediately threatened, but those species that are sort of the tier right after that. And so they're perhaps not so rare, but may start to be susceptible to abrupt changes. And so those are perhaps species that aren't even yet of interest to management, but quickly might become of interest to management. And so we want to start highlighting and building this idea in the sort of early warning and forecasting next step of our project. So hopefully that sort of answers that question. I don't know if you wanted to add anything.
Yeah, I just wanted to add, Allison pulled a question from the YouTube live stream that kind of sort of gets to the same thing. Someone was asking about, how important then is it it sounds like from your presentation that it's really important to record common species, right? So not just nudibranchs, right? Or just like more rare, sparse, charismatic things. So they're asking if that's true. And also so I'll answer that part first. And so yes, when we put together our Snapshot Cal Coast, like most wanted species guide, we've just started last year actually including like a list of species that we really want people to look for, things that we know that have like management, that are management driven questions, like is this species moving north? Is this a new invasive species, et cetera. But we also added a checklist to remind people to take pictures of common things, especially things like barnacles and mussels, so maybe like the less charismatic common things, so not just sea stars and sea anemones. And this, I think, kind of gets to the kind of data we need, like a more comprehensive picture. Like ideally, when we go out there or one goes out there, you can take a picture and try to document every species you can find. And so that includes like seaweeds you might not be able to tell apart, or barnacles that you might not be able to tell apart. And that gets to the second part of this person's question was asking, is it important to record common species of which you feel certain of the ID? And the answer is yes and no. Like, it's important to take species of everything, but you don't need to know the identity or the name of the species you're taking a picture of. What you need to do is take a good enough picture or set of pictures that the combination of the machine learning on iNaturalist and the community can help put an identification on that observation. You don't have to know what you're taking a picture of. Hopefully you start to learn the things that you're taking pictures of, but you don't have to know. And I can also just ask, like, use this to answer Melissa Miner's question, which was about how are species IDs confirmed and vetted, and the community helps confirm the identification of observations. And that process is dynamic. So if something is vetted and becomes research grade and is shared with GBIF, as Gio described, that identification can still change if someone else or the community goes back and looks again and says, well, actually, maybe that's not the species these people thought it was. And so I encourage anyone with taxonomic expertise who's interested in using these data or understanding these data more is to get on iNaturalist and start making some identifications of taxa that you know, and start to see how the process works. And it's important to remember that these data are like any other data source. Like, they need filtering, like Gio was saying, and they need curation. Like, every time Allison and I go to answer a question about a species of crab, for example, and we dig into the iNat data, we end up re-identifying hundreds of observations because we're looking more closely. And this is the same thing that happens for me when I go into the museum collections and dive deeper into everything in jars that's labeled as one species. Is you have to look and examine it more closely, and you make lots and lots of changes. And so one of the best things about iNat data is there's evidence that we can go back and re-examine and make those changes. All right, so I'm going to stop talking. We can go on to the next question.
Where are my questions? Okay, so we'll take one last question. I apologize to you if I'm not answering your question. I'm prioritizing the sort of non-iNaturalist specific questions. But so I'm going to take this one from Ryan Mayer, who's asking about the importance of snapshot CalCoast in the overall picture that we're getting from iNaturalist, and how much snapshot CalCoast contributes to iNat activity, and would it be possible to do all this without it? And I think for me, in my experience, and I'll let everyone else speak after that, but it's the answer is a resounding no. I mean, we've seen, I've showed you the pulse and the peak that you get from snapshot CalCoast. But more than that, it's not just the number of observations, it's really the complexity of the number of species and what is being documented and how comprehensively and how many spots around the coast. And so that picture is invaluable. And if you go on the Places app, you'll find that some places, a decent amount of MPAs are grayed out. They're not available. And they're not available because there aren't enough data. And so we are working on adding those and guiding people towards going there if it's possible to make iNaturalist observations relatively easily there. But certainly we probably would have way more grayed out MPAs if it wasn't for snapshot CalCoast and the sort of targeted effort to get people all over the coast, and especially in MPAs. So I don't think any of it would be possible without snapshot CalCoast. I don't know if you want to add anything else to that. Otherwise, we're out of time. All right, that sounds like a no. It's all good. So thank you so much for joining us today. And I hope you continue to engage with us. I hope you go out and play with the tools and read the report when it comes out. I'll be sure to publicize it with everyone. And please keep in touch and just reach out to us if you're interested in finding out more or collaborating with us. But it's really appreciated. I know it was a relatively chunky webinar today, but you know, hopefully you enjoyed it and took something away from it. So thanks so much, and stay safe. And yeah, thanks so much for joining. Thank you.
Get out to the coast and make observations if you can.
Oh, right. Yeah. Snapshot Calcoast 2020. Please join us. Yeah.
And we'll be sure to send information to all attendees of today's webinar.
Yeah. Any follow-ups and the recording, in fact, if you wanted to watch it all again.
Thanks, everyone.
Bye, everyone.
Thank you, everybody.
Woohoo. Can I stop the recording, Gio?
Sweet. Let me stop the live stream. Bye, YouTube.